All right, everyone, we are live. A very good afternoon and warm welcome to Shri Chaitanya. This is your NEET 10, a game changer series in which we are going to study for 10 days, which are the last lap for our NEET 2024 exam. So NEET 2024 exam will be on 5th of May. So on daily basis, we are doing 25 questions from 25 different concepts of 11th and 12th. So in today's session, again, we are going to do 30 questions today. I am having 5 bonus questions for you people as class was not there yesterday. So these are 30 different concepts basically, and these concepts I have picked up from previous 10-year question papers of NEET. So all those concepts which are repeatedly being asked into your NEET examination, I have picked up all those topics and framed questions from those particular topics, and we are going to solve all those questions alongside understanding all the topics which are there. Okay? Yes, good afternoon. So first thing first, everybody, do hit the like button and share it with all your friends so that everybody can participate. We can solve the question in a better frame of mind with best of synergy. All right, good afternoon, Sefwan. Good afternoon, Avinash. Ripak, very good afternoon. Uday, hello. Yes, please. What is your doubt? Do tell me. Uday Kumar, you can ask your doubt. Come on, what is your doubt? All right, so without any further delay, let's get started for today's session. So this is Ultimate Series for NEET 2024, which claims that you're gonna score 650 plus marks from these sessions for 10 days, and we are having session for all the 4 subjects, be it chemistry, be it physics, be it botany and zoology. This is me, Beena Yadav, your botany mentor. We will be having class on botany. After that, Pawan Kasu sir will be taking your chemistry sessions from 4:30 to 5:30 p.m. today itself. All right. Avinash, ma'am, give tips to solve assertion and reason question. See, Avinash, while solving AR type of question, nobody is very sure about first and second option basically, okay? Everybody is quite sure about third and fourth option. Am I right? Yes. You got confused between first and second option. Because both the statements are correct, that's where the crucial part comes in. That's where you think, yeah, reason is correct explanation or not. To understand this thing, just use this one experiment, okay? While solving AR type of question in today's session, use it as a trick. Read out the assertion statement loud out, okay? Put a big question mark in front of it and seek for the solution. What should be the exact explanation of this question mark? If that exact explanation is there in the reason, then option 1 or option 2. As simple as that, okay? Yes. So that's how you can solve AR type of question. Option 3 and 4, in which your assertion is incorrect and assertion is correct, reason is correct. That is very simple to decode. Basically, the problem arises between first and second option. Yes. So try this trick out in today's session only. We will be solving AR type of question. Try this trick and then let me know whether it has worked for you or not. All right. So without any further delay, kids, let's get started with our very first question. On your screen, very first question, everybody, on your screen, the question is about cyclosporin A is used for. Question is from microbes in human welfare chapter. And repeatedly, if you will see PYQs from cyclosporin A or let's say statin, every year question has been asked from this particular topic. So cyclosporin A is used for what? I can see the answers. Everybody, please participate in the session. Everybody do answer in the comment box. Avinash says option C. Rebak says option C. Munni says option C. Rest of the kids, come on. Anjana Reddy says option B. All right. Rest of the kids, come on. Come on. Try your 100%, give 100%, try your level best, everybody. B and C. Okay, I'm getting answers between B and C. So cyclosporin A is basically used for immunosuppression, not for removal of blood clot, not for lowering down blood cholesterol level or lowering blood sugar level. It is used for immunosuppression. All right, so correct answer will be option number C. Yes, Anjana? Yes, Rebak? Yes, Munni? Correct answer has to be option number C. Perfect. Let's move to the second question then. Next question, kids, on your screen. Very important question. Try to solve the question. Second one, bachcho, on your screen. Yes. Avinash has answered. All right. Rest of the kids. Uday Kumar, yes, hello. Please ask your doubt. Rest of the kids, come on, come on. Answer, answer. The most abundant prokaryotes useful to humans in making curd from milk and in production of several of antibiotics are described based on their mode of nutrition as. Based upon their mode of nutrition as. Rebecca has answered. Vanama Vandana, good afternoon. Good afternoon. Please solve the question. Yes, yes. The most abundant prokaryotes useful to humans. If I speak about prokaryotic organisms, okay, so in terms of nutrition, they have maximum nutritional diversity. What do I mean by that? What do I mean by maximum nutritional diversity? Okay, nutrition wise, any organism can be autotrophic, number one, can be heterotrophic, number two. In case of autotrophic, organism can have photoautotrophic mode or it can have chemoautotrophic mode. In terms of heterotrophic nutrition, we can have either saprophytic, we can have holozoic nutrition, okay, we can have parasitic mode of nutrition. So you visualize any kind of nutrition, everything is found in group of bacterias. Okay, so bacteria or the prokaryotes are nutritionally diverse. They do photosynthesize, okay, that energy can be taken up from sunlight, energy can be taken up from chemical reactions, but yes, they can prepare their own food and they can be dependent upon different organisms also for the requirement of food, all right. So we can see maximum nutritional diversity in case of nutrition. But there is a big but here. The reason is your bacteria, the vast majority of the bacteria are heterotrophic. That's what your NCERT says. Okay, although we can have all the possible options, but majority of them are heterotrophic in nature. So the question is asking about the most abundant prokaryotes useful to humans in making curd. When we want to make curd from the milk, basically we need to add inoculum to our milk, and that inoculum is nothing but lactic acid bacteria only. Okay, that is nothing but the curd only. So this curd is basically, the lactic acid bacteria present into the curd is basically multiplying. After multiplication, that is responsible for coagulation of the milk. When your milk got coagulate, it forms curd. All right. So coming back to our question, basically the vast majority of the bacteria are heterotrophic in nature. Okay, your NCERT says the vast majority are heterotrophic. All right. So correct answer will be option number D, +4. Can I say +4, Savinash? Rebecca? Everybody +4? Yes. Is that +4? Perfect. Moving on to the next question then. Next one on your screen, kids. Pick up the incorrect statement. So the key word in this question is incorrect. Do underline the keyword. Okay. We need to find out the incorrect statement out of these 4 statements. Very interesting question. Start solving from option number 1, which says, in diatoms, the cell wall forms 2 thin overlapping shells which fit together as in a soap box. Yes, of course. If I speak about chrysophytes, specifically diatoms, we have 2 thin overlapping shells which perfectly fit together, just like a petri plate or a soap box. All right, this is a correct statement. Clear. Then fungi shows a great diversity in morphology and habitat. Yes, of course, in fungi we have many type of fungi which has got different morphological behaviors, and their habitat is little different. Okay, this is again correct. Now let's see option number D, which says mycoplasma is the smallest living cell that can survive without oxygen. Yes, of course, mycoplasma, or we can call them PPLOs, pleuropneumonia-like organism. They do not have cell wall that makes them smallest of all the living organisms. All right, so this is perfectly correct statement. Then we have claviceps that is used extensively in biochemical and genetic work. Is it claviceps or is it neurospora? Yes, neurospora. Yes, kids, it is neurospora crassa that is used extensively for biochemical and genetic work, which makes this statement incorrect, hence correct answer. Okay? Yes, it has to be neurospora crassa. Perfect. Can I see +4s in the chat box? Rebecca, Vanama, Avinash, +4s. Come on, kids, +4s in the chat box. +4, +4, +4, +4. Great. Moving on to the next question. Next one, this is from Plant Kingdom. Which one of the following shows isogamy? Dear kids, pay attention. Isogamy with non-flagellated gamete. First of all, what is isogamy? Isogamy simply means your organism is having iso means same. Gamy means gamete. So your organism is having same gamete, which means same in terms of size, in terms of shape, in terms of motility. If the gametes are motile, both of them are motile. If the gametes are non-motile, both of them are non-motile. Okay? So in terms of motility, in terms of size, shape, they are perfectly similar. Those ones are called as isogamy. Now the question is asking you about isogamy, which means in terms of size and shape, your gametes are similar, with non-flagellated, which means my gametes are non-motile. My gametes are non-motile. Okay? Is it a case of sargassum? Is it a case of ectocarpus, eulothrix, or spirogyra? Your NCERT clearly mentions it. Okay, your NCERT has given you example of isogamy with flagellated gamete as well as isogamy with non-flagellated gamete. All right? Yes, it is your option number D, which is spirogyra. Which is spirogyra. Okay, you people are confused between flagellated and non-flagellated. See, isogamy means both the gametes are perfectly similar in terms of size, in terms of shape. Everything is similar, but if they both are motile, we call them flagellated gamete. If they both are non-motile, we call them non-flagellated gamete. Okay, Nagaraju. Okay, is it 0 for you? People, +4 or -1? Come on, students. +4, -1 or zeros? +4, -1 or zeros? 0 for Nagaraju. Avinash, is it +4? -1, not a problem, not a problem. All right, perfect, perfect. Moving on to the next question. You can redeem yourself in this next question. Okay, meanwhile, if you are liking the session, then do hit the like button also and share it among all your friends. Yes, next question. Select an incorrect match. Incorrect, this is the key word here, incorrect match. Okay, so the first one is actinomorphic flowers. Example, datura. Second is zygomorphic flowers, example, pea plant. Third is asymmetrical flowers, example is canna. Then we have hypogynous, example is cucurbits. Okay, so basically our flowers are great answers, Avinash, Nagaraju, Murli, perfect. Our flowers are segregated based upon the symmetry of the flower. Okay. See, my flower, I cannot cut my flower into 2 equal half by any plane. Such kind of flower is called as asymmetrical flower. Whereas, if I am able to cut my flower into 2 equal half, I call that symmetrical flower. So, my symmetrical flower can be of 2 different types. We can have actinomorphic, we can have zygomorphic. Okay, we can cut my flower into 2 equal half, but by how many plane? if I'm having more than one plane in which I will cut my flower into two equal halves, that's what we call as actinomorphic. But if we have just one plane in which I'm able to cut my flower into two equal halves, that is called as zygomorphic, okay? And asymmetrical is the one which cannot be cut into two equal halves, right? So we have actinomorphic, asymmetrical, zygomorphic, then we have hypogynous. We need to find the incorrect match. Everybody is correct here. Avinash is correct, Murali is correct, Murari is correct, Vanama is correct. So basically, your hypogynous flowers are the ones, see, gynous means ovary, all right? Hypo means below. So this word basically resonates with the position of rest of the floral parts with respect to ovary. So rest of the flower parts with respect to ovary are below in the position. So my ovary is above it, which means my ovary is having superior position. So the superior ovary is found in the case of hypogynous flowers. Okay, this is incorrect match, hence correct answer. Right? Yes, superior ovary will be there in case of hypogynous flower. All right, everybody, +4 or -1. Come on, let's move to the next question then. Identify the function of the part labeled as A from the diagram. This is our diagram and this part is A. This one is A. This part is A. We need to find out the function of this particular part. Come on, tell me. Function of this particular part. Yes, kids. Yes, yes, kids, yes, yes. Shriman, okay, Vanama, Avinash, Murari, great answers. So, see, this is the area of meristematic activity. This is my elongation area, and finally, this one is my maturation area, right? So, this is having area of meristematic activity, above which we have the area which is for elongation. So, it helps to increase the length of the root because increased vacuolation is there, and that will help my root to increase its size basically, okay? And over here, this is my area of maturation, wherein the permanent cells are present and they do have root hairs also. Root hairs are basically to increase the surface area for water absorption. Okay, due to presence of these root hairs only, we are able to increase the surface area so that my plant can absorb more amount of water and minerals. Right? Yes, perfect. Moving on to the next question then. Next question, kids. Plant linear growth is indefinite mainly because of, in plants we can say the growth is indefinite. For infinite period of time the growth is happening. That is because of a specific type of tissue that is found in the case of plant that we do not have, okay? Unlike us, a plant have, the growth of the plant is for infinite period of time. Very good. Avinash, good. Murari, very good. Vanama. Rest of the kids, come on, come on. That is indefinite mainly because of the presence of apical meristem. So in case of plant, we have shoot apical meristem and a root apical meristem. At tip position of the shoot, we have meristematic tissue which is continuously dividing. Okay, if my tissue is in continuous state of division, which means it will not perform any specific function throughout the lifespan, it will just divide. So due to the presence of apical meristem on both the places, shoot and root, the growth of the plant is for indefinite period of time. Alright, so answer has to be option number B, which is apical meristem. Is that correct, everybody? Is that correct? Can we have +4? Can we have +4 in this one? Yes, +4 everybody. Moving on to the next question. Which tissue are responsible for the plant's flexibility? Okay, we have leaf of the plant. They are able to flutter in the wind. Okay, due to the presence of a specific tissue, they are able to flutter in the wind. What is that tissue that is providing mechanical strength, and it is a living tissue also? Come on, it is a living tissue and providing mechanical strength. Murari, Vanama, Avinash, great kids. It is basically your colon-chyma tissue. So my colon-chyma tissue is living mechanical tissue, kids. It is my living mechanical tissue. It provides flexibility. Okay, so correct answer will be option number 1, which is colon-chyma tissue. +4 in this question. Yes, Ayushi, can we have +4 in this question? Moving on to the next question. This is from anatomy, secondary growth. The sapwood occupies which area? The sapwood. Let me be on the side. You will be able to see the question properly, everybody. Yes. Sapwood. Come on. Murari has answered. Vanama has answered. Come on, kids. So basically, if I speak about secondary growth, Ashrita has answered. Avinash has answered. See, if I speak about secondary growth, the centermost part in which we have too many secondary xylem, in which we have all the secondary metabolites also, there is no space for conduction of water and minerals. So the centermost part, which is hard, which is durable, which is resistive to microorganisms, and this part is no longer be participating into conduction of water and mineral. So that centermost part is called as heartwood. Okay, now the main function of xylem is conduction only, and this centermost heartwood is not able to conduct. So basically, the surrounding tissues from the heartwood, the peripheral ones, they are basically participating in conduction of water and minerals. So these peripheral ones are called as sapwood. Okay, so the peripheral area of the secondary xylem and is involved in the conduction of water and minerals from the root to the leaf. So heartwood is hard, durable, resistive to microorganism, cannot participate in conduction of water and mineral, but your sapwood, which is in the peripheral area of the heartwood, is actually participating in conduction of water and minerals. Is that correct? Yes, everybody? You understood? Yes, can we have +4 in this question then? +4 kids, I can see almost 80% of you have done it correctly. Yes? Perfect. Moving on to the next question then. With a +4, let's move to the next question. We have this diagram. Identify the cell organelle and select the correct match. What is this organelle? And we need to find the correct match with respect to this organelle. Okay, the first option says it is chloroplast and the function is ATP synthesis. The second option says chloroplast and the function is photosynthesis. Third option says it is mitochondria. Okay, mitochondria found less in number in metabolically active cell. Option D says mitochondria and inner membrane contains enfoldings that help in increasing the surface area. First of all, when we see this diagram, of course, this is not the diagram of chloroplast. This is my mitochondria. In case of mitochondria, this is my outer mitochondrial membrane. Alright, whereas this one is my inner mitochondrial membrane. We can clearly see inner mitochondrial membrane has got enfoldings, which are these ones. These ones are called as cristae. Yes. So cristae are nothing but the inner mitochondrial membrane's enfoldings. Okay. So it is not chloroplast. It is mitochondria, yes, but in case of metabolically active cells, wherein my cell is in continuous state of division, we do have more number of mitochondria, not less number. Okay, so this is again incorrect, and this is the correct one. Mitochondria, inner membrane contains enfolding, which is called as cristae. Good afternoon, Naju. Good afternoon. Come quickly. Yes, so the correct answer has to be option number D. Is this correct, everybody? +4, Revak? Plus 4. Ayushi, plus 4. Plus 4 kids? Yes? Alright, moving on to the next question then. Next one. Non-membranous nucleoplasmic structure in nucleus are the site for active synthesis of what? Baba Kharif. Yes, yes. There is this one non-membranous structure inside the nucleus. Okay, that is the site for active synthesis of. Do we synthesize protein? No. Do we synthesize mRNA? No. tRNA? Oh, please, my dear kids, don't get confused with this one term, non-membranous nucleoplasmic structure. There is this nucleoplasmic structure, which means inside the nucleus it is present. Inside the nucleus we have got presence of nucleolus. Okay, don't get confused everybody. Yes, just by looking at non-membranous, don't answer the question. It is non-membranous and it is nucleoplasmic structure also found inside the nucleus. Okay, so inside the nucleus we have nucleolus responsible for active synthesis of our RNA. Option number C. Maximum of you have answered option number A, that is protein, because you thought membraneless is ribosomes only. Just by looking at this one word, which is membraneless, you just thought it is ribosome. Hence, you answered option number 1, which is protein. Am I correct? That's how you have answered? Am I correct? Yes? Yes or no? Am I correct or not? Tell me. Yes, ma'am, you are absolutely correct. So here comes a very important lesson for all of you. When you are writing your exam of NEET, please make sure every presumptions that you do have, keep it aside. Start reading the questions afresh. Start develop a habit to read complete question. Develop a habit to read all the options. Carefully read the question, carefully read all the options out there and then answer. Okay, just by saying one option you cannot answer, because that will give you -1. You will lose 4 marks of the question and that will also give you negative marking. Alright, so please make sure while solving the questions, try to read the complete question. Not try, you have to read the complete question. Okay, and all the 4 options also. Will you do that for me? Yes? Will you do that for me? Yes? Is that a promise? Kids, is that a promise you will do that? Yes, ma'am, we will definitely do that. Okay, perfect. Moving on to the next question then. Next question. Identify the correct statement with regards to G1 phase of interphase. So interphase, basically we have G1 phase, we have S phase, we have G2 phase, and then finally we have M phase. Okay. So this is the sequence which myself follows. G1, S, G2 and then finally M. Alright. So basically for G1 phase, we need to find the correct statement. What is happening inside the G1 stage? I can see lot many correct answers. Avinash, Murari, Vinama, Ayushi. Everybody please keep on answering. It is basically your cell is metabolically active. It grows but does not replicate its DNA, because replication is happening in S phase. Replication is there into S phase. Okay, so the correct answer will be option number 2, which is cells are metabolically active, they are growing but they are not replicating its DNA. Alright everybody, moving on to the next question with your +4 and -1s. Next question on your screen, come on. Next one, a bivalent of meiosis 1 consists of bivalent. What is a bivalent? What is a bivalent? Bivalent is basically your homologous chromosomes. This is your bivalent, right? A pair of homologous chromosome is called as bivalent. Yes, a pair of homologous chromosomes is called as bivalent. Now, this bivalent, wherein we have these two chromosomes, we have 1, 2, 3 and 4. We have total 4 chromatids. Yes or no? 4 chromatids. And we have this 1 and 2 centromere. Alright, so answer will be we have 4 chromatids and 2 centromere. Centromere is basically the primary constriction joining your chromatids. Alright, so we have 2 chromatid per chromosome. Since we have 2 chromosomes, we are having 4 chromatids. That is why the condition is your tetrad in terms of chromatids and bivalent in terms of chromosomes. Alright, so answer will be option number C, 4 chromatids and 2 centromere are there. Alright, moving on to the next question then. Next question, kids, come on, solve the question. The bonds representing the label A and B respectively are. So we have this A which is there, we have over here B. Okay, so the bonds representing the labels A and B, what are they? Your A is hydrogen bond. We can see this is a bond between thymine and adenine, right? And this is a bond which is joining nitrogenous base with sugar. Very good, very good, very good. Murari, Avinash, Vanama, Rebak, great answers. So this is basically, this is your hydrogen bond and this is your glycosidic bond. Everybody is correct. It is option number C. Okay, perfect. Perfect. See, your okay. Somebody is saying option A also. Listen to me very carefully. This A option is basically a bond between thymine and adenine. We know it very well. Your adenine and thymine, they are bind with 2 hydrogen bonds, whereas your guanine and cytosine, they binds with 3 hydrogen bonds. Alright, and the glycosidic bond, this one, glycosidic bond, is basically joining your sugar with nitrogenous base. Okay, that is why A is hydrogen bond and this B is N-glycosidic bond. Clear now? Yes. Perfect. +4. With your +4, let's move to the next question, kids. Next question, little bit lengthy question, but it is an important and interesting question. And everybody, please do hit the like button also. Do hit the like button. It motivates us also, okay, and share it with all of your friends. Such kind of sessions are, we are taking such kind of sessions on daily basis, so do hit the subscribe button also and press the bell icon, okay? Come on. As glucose is prepared at the source by photosynthesis, it is then moved into, glucose has been prepared, after that it will be moved into A cell and then into, from A it will move to B cell by some process. We need to tell what is this A cell, what is this B cell and what is this process which has been involved. Glucose has been synthesized, after which it will be moved to some cell which is going to transport it, and from that some cell moved into some another cell by which process? Come on. You got the question, everybody? Yes. So basically we are discussing mass flow hypothesis here. If you have to transfer your phloem sep, very good Avinash. I can see the answers. Ayushi, great. Please try your level best. Try your level best, everybody. What is this A? What is this B? What is this C? Okay. So if my plant has synthesized glucose molecule, right, this glucose molecule will be given to my companion cells. Okay, my sieve tube cells are basically enucleated. They do not have nucleus. Nucleus is basically brain of the cell. So in turn, they do not have brain. If they do not have brain, they cannot move our phloem sep into rest of the tissues. That's the part where companion cells came into the picture. Companion cells are basically directing the flow of phloem sep with the help of sieve tubes. Okay, so at first at first, my dear kids, they will be gone into companion cell, and then companion cell will move. Okay, companion cell will move it to the sieve tube cells, and then finally through active transport they will be transported to the rest of the parts. Okay, yes, okay. Okay, some of you might think that this part is from transport of plant, but my dear kids, from angiosperm's perspective, your examiner can definitely ask such type of question. See, the syllabus which has been reduced, basically somewhere you will feel it is interconnected to the few topics which are already there. So wherever you find that interconnection, please do read all those concepts, please do read all those topics, because your examiner can ask them. Okay. Okay, since angiosperms are still there in your syllabus, the examiner can ask about such kind of process involving sieve tube and companion cells. Okay, yes, yes. All right, everybody. So with +4 -1, let's move to the next question. Next one is choose the incorrect match. Incorrect. Pay attention. We need to find incorrect match, right? Then, with respect to the photosynthesis, let's try to read each statement one by one. Yes, Hanji. First is thylakoid of chloroplast. Thylakoid. They are responsible for light reaction of photosynthesis. Indeed, thylakoid has got presence of all the pigments which are there, and the pigments are responsible to absorb a wavelength of specific light. So of course they are participating in ATP formation. Number B, reaction center of photosystem 1 is at 680 nanometers. You need to brainstorm that PS1, we have photosystem 1 and photosystem 2. In photosystem 1, the absorption maxima peak is at 700 nanometers. In 680, is it at 680 nanometers? Okay, but the question says the reaction center of photosystem 1 is a 680. That is incorrect. All right, so option number B is incorrect. Avinash, 720 marks you are aiming for your NEET exam, not for photosystem 1. Okay, I understand. The figure of 720 is basically has stuck into your brain because we want to get 720 out of 720. Is that true? Yes. Okay, perfect. So reaction center of PS1 is basically at 700 nanometers, not at 680. This is incorrect statement, right? Option number C, Cohn Van Neel experiment on purple and green sulfur bacteria, perfectly correct statement. Then chlorophyll B, component of light harvesting complex of photosystems. These are correct statement. Incorrect is option number 2, and question has asked about incorrect option. That is why answer is option number B. Clear, everybody? Clear? Yes? Okay. Can we have +4? +4 in this question? Kids, can we have +4 in this question? Yes, ma'am, we can have +4. It was an easy question. Yes, Royal? All right, moving on to the next question then. Next question, what is the ratio between the number of CO2 involved versus ATP needed versus NADPH required, respectively, for successful completion of 1 turn of Kelvin cycle? So if we speak about C3 cycle, 1 turn of Kelvin cycle, question is asking about per CO2. If I want to fix 1 CO2, how many ATP will be required? Okay, and how many NADPH will be required? So per CO2 fixation, dear kids, 3 ATP and 2 NADPH2 is required. Okay, so per CO2 means 1 CO2, 3 ATP and 2, which comes out to be 1, 3 and 2. Okay, since we need to fix glucose molecule, so we have to multiply this with 6, because glucose molecule has got 6 carbon dioxide. All right, Yes, so it is 1 is to 3 is to 2. 1 is per CO2, 3 is the number of ATP, and 2 is NADPH2, right? 1 is to 3 is to 2. Clear, everybody? Yes. Yes, 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 we are clear. Bhima, Rebecca, Avinash, Sanjana, Vanama, Yashashwini. Is this clear? SJ, Ayushi. Is this clear? +4? Okay, perfect. Moving on to the next question then. Next question, dear kids, on your screen. All are true about glycolysis except. So we have 4 statements. Basically, we need to find the statement which is incorrect. Okay, the question is asking you about. We have all are true about glycolysis except. So we have 4 statements. 1 is incorrect. Rest 3 are correct. Uma Devi, that's such a huge compliment, bache. Thank you so much. Thank you very much. Please solve the question. So glycolysis, it is a 10-step process. Yes or no? Okay. Glucose is phosphorylated to give rise to glucose 6-phosphate. We have added phosphate to glucose molecule. That is why it is glucose 6-phosphate. By the activity of hexokinase, the very first step, okay, it is perfectly correct, perfectly correct. Reduces 2 molecule of NAD plus for 1 glucose molecule. Per glucose molecule, we have 2 NADH2 which are produced, right? Perfectly correct. It produces 1 pyruvic acid molecule from each glucose molecule. No, no, no. Per glucose, we have 2 pyruvic acid, okay? If I speak about glycolysis, 1 glucose will give rise to 2 pyruvic acid. It will give us 2 ATPs. It will give us 2 NADH2. Okay, so 4 ATP are produced, 2 NADH2 are produced, and 2 pyruvic acid has been synthesized. But 2 ATP has been used also. So net gain will be 2 ATP, 2 NADH2, and 2 molecules of pyruvic acid. Okay, so correct answer will be option number D. It is incorrect statement, hence would be correct answer. Instead of 1 pyruvic acid, it has to be 2 pyruvic acid will be synthesized. Alright, 2 pyruvic acid will be synthesized. Is this clear? Yes, everybody? Geetika, Sanjana, Umadevi, Avinash, clear? With your +4s, let's move to the next question then. Next one, kids, on your screen. Come on. The reaction of Krebs cycle takes place in. What is the location? Okay. So if I speak about number 1 process, which is called as glycolysis, it is taking place in the cytoplasm. If I speak about link reaction, the location is matrix of mitochondria. If I speak about Krebs cycle, again the location is matrix of mitochondria. If I speak about electron transport chain, the location is inner mitochondrial membrane. Alright, so the question says the reaction of Krebs cycle takes place in 2. In cytoplasm, we have glycolysis, dear kids. No relation to endoplasmic reticulum. On the surface of mitochondria, specifically into inner mitochondrial membrane, we have electron transport chain. So correct answer has to be option number C. Alright? Okay, perfect. Moving on to the next question then. Answer is option number C. Moving on to the next question, kids. Which one of the following plants does not show plasticity? Okay, what is plasticity first of all? Then we will think about which of these examples does not show plasticity. So basically, if I speak about the final product, okay, if my plant is using different pathways in different condition, then obviously the product will be different. Okay, for the same process, if I have chosen different pathway, the product will be different, something called as plasticity. The very common example is the shape of the leaves, called as heterophyly. Heterophyly, if you will carefully see coriander leaves, so the juvenile coriander, the immature, okay, coriander is having different shape of leaves than the mature version of it. Okay, it goes same with buttercup and cotton. So for heterophily, the examples are coriander, buttercup and cotton. Maize is not the example. Okay, and question was asking about does not show plasticity. That will be my maize. Maize will not show heterophily. Maize will not show plasticity. Okay, so answer is option number C, which is maize. Yes, Vanama, Sanjana, Rebecca, Avinash, Yashashvini, Geetika. Is this clear? Niranjan. Hi, yes, of course, I can explain it one more time for you. Niranjan. See, if I speak about heterophily, heterophily is basically presence of different type of leaves. Okay, coriander is one plant, but we are able to see different type of leaves on coriander. How come? Because there is a process called as plasticity. What is plasticity? If let's say there is formation of leaves, if there will be different pathway followed, then the shape of the leaf will be different. If you will see coriander, which is little young, little in the juvenile phase, the shape will be little different than the mature version of it. Okay, so this shape of leaf, which is called as heterophily, that hetero means different, phily means leaf. So we have different shape of the leaves. That is because of the plasticity. Okay. Yes. So correct answer is option number C, which is maize. Niranjan, I hope it is clear to you now. Let's move to the next question then. Geitonogamy involves what? Bhima is asking me to explain differentiation. See Bhima, in the callus we have all the tissue which is basically into the meristematic state, which is in continuous state of the division. The cells are basically not permanent, they are not doing any permanent function, they are just dividing. Alright? Let's say if my cell leave the function of division, let's say if my cells leave the function of division and start performing a specific function, I call that permanent cell. Okay? So this phenomena wherein my meristematic cell called as undifferentiated cell has participating into a specific function called as permanent cell, this phenomena is called as differentiation. Okay? So my undifferentiated cell, meristematic cell, is converted into a differentiated called as permanent cell. This phenomena is differentiation. Okay? Uma Devi, I hope it is clear to you now. Yes, kids. Come on, solve this question. Come on, solve this question. Come on. Yes. Option A, all right. Rest of the kids, come on. Geitonogamy involves what? Okay, let me give you a simple clarification about three different type of pollination which is there. Okay, Shri Devi, option D. We have three different types. One is autogamy. Second is geitonogamy, which was asked in the question. Geitonogamy. And third, my dear kids, is genogamy, or we can also call it allogamy, one and same thing. Okay, so we have these three different types, right? So if I speak about autogamy, basically in case of autogamy, the pollination is in the same flower. Obviously this same flower is of same plant. Okay, if the transfer of pollen grain is within the flower itself, we call that autogamy. Okay, but if the transfer of pollen grain is in the different flower, in between two different flowers, but these two different flowers are basically on the same plant, if they do belong to the same plant, they both belong to same plant, that's what we called as geitonogamy, right? But my two flowers in which pollination is happening, these are different flowers, but these two belong to two different plants. that's what we call as genogamy. Okay, so my autogamy is purely self-pollination. Okay, there will be no variation at all. My geitonogamy, since the flower belongs to the same plant, there will be no variation. So if I speak about genetically, this is similar to autogamy, genetically, because there will be no pollination. Sorry, there will be no variation. Okay, but if I speak about functionally, so since the pollination is within the flower, we do not need any external agency to transfer my pollen grains, right? In this case, since the pollination is in between two different flowers, we do need outside agency in order to transfer my pollen grain from one flower to the another one. Similarly, I do need agents in this case also. So, if I speak about functionally, it is similar to genogamy. Okay, so genetically my autogamy, geitonogamy is similar to autogamy, but functionally it is similar to genogamy. Is this clear? Yes? Okay, let's come back to the question. Geitonogamy involves what? It involves fertilization of a flower by the pollen from another flower of the same plant. Okay, so for the same plant we have two different flowers in which pollination is happening. Clear everybody? Can we have +4? Yes, +4. Okay, then next question on your screen. Yes, next question kids on your screen. Come on, and hit the like button also. Come on, hit the like button also. Among the following, which one correctly represents the monohybrid test cross? Monohybrid test cross. What is test cross first of all? Supposingly, if I say the parent is having tall height, but by tallness, I do not know that this tallness is because of homozygous genotype or heterozygous one. Okay? Yes, for that purpose, we need to do test cross. What is test cross? Test cross is crossing of the parent with its homozygous recessive form. So, if I am crossing my heterozygous parent, which is this one, with its homozygous recessive form, which is this one, then the ratio will become out to be 1:1. Okay, this is my test cross. Getting the answers? Great, Sanjana. Yashashwini, Vanama, Rebecca, very good. Naju, yes, it is live on Goshala also, live on our parent channel also. Great, great. It is cross between F2 tall and dwarf parental plant. That's the correct answer. Okay, perfect. +4 everybody. Moving on to the next question. Next one on your screen. Which of the following acts as inducer for the expression of lac operon? Okay, for lac operon, for the expression of lac operon, which is acting as inducer. So basically we have I, Z, Y, A. Okay, this is a regulatory gene. Regulatory gene. Right, this will code for beta galactosidase, this will code for permease, this will code for transacetylase. Okay, so the product of I gene will basically attach to operator, and if it will attach to operator, my RNA polymerase will not be able to bind to promoter. If RNA polymerase is not able to bind, it will not be able to go for transcription process of Z, Y and A gene. Yes? Yes, so for that purpose we have an inducer called as lactose that will bind with the product of this regulatory gene, hence stopping it for joining to operator so that my RNA polymerase can join to promoter and go for transcription process. Okay, so this is the inducer is basically my lactose. It is lactose. Correct everybody? Yes, correct. +4. +4 in this question, Sanjana, Vinama, Avinash, Rebecca, Yashashwini, everybody +4. All right, moving on to the next question. A student was asking me about AR type of question in the beginning of the session. Please come on, solve this question. We have assertion, we have reason. Assertion says, in nucleosome, histone proteins are basic in nature. Now in reason, you need to find out that why this basic nature is there. If you got the answer of why this basic nature is there, then you can have answer option number 1 or you can have answer option number 2. Okay? Your question is saying, in nucleosome, histone proteins are basic in nature. What contributes to their basic nature? What contributes? Jignesh. See, DNA is acidic in nature. That is a separate story altogether. We are asking about histonic protein. Why they are basic? Reason being they are rich in 2 amino acids called as lysine and arginine. That makes them basic in nature. Okay? That is why both are true and this is correct explanation also. Yes, Jignesh? Yes, Avinash? Rest of the kids, did you got it? So, similarly in all the AR type of questions, try to find the answer. See, in this question, in assertion statement, my statement says histone proteins are basic. Why basic? If you know the reason why in this statement, then answer option 1. Or if you don't understand the reason behind it, then answer is option number 2. Okay, I hope it is clear to everybody. Answer is option number A. Moving on to the next question then. Next one, kids. And please, I hope you like this session. These questions are really important from very different topics. So please do hit the like button also and share it with all your friends and subscribe to our beautiful channel. Come on, come on, come on. Which nitrogen base containing nucleotides do not polymerize in PCR? Which nitrogen base having nucleotides do not polymerize in PCR? So in polymerase chain reaction, my DNA can polymerize, right? My RNA cannot. In DNA, we have, yes, great answer. Rebecca, Avinash, Vanama, Vandana, Sanjana. Yes, my DNA do not have uracil. It do have thymine, do have adenine, do have guanine, but uracil is not there. Right? So answer will be option number C, which is uracil. Great, everybody. +4 and next question. Next question. Which of the following are referred to as molecular scissors? Molecular scissors are basically the ones which are able to cut in between the molecules. Okay, if I need to cut nucleotide in between the molecules, I would call that molecular scissor. Now, which enzyme behave as molecular scissor? Great answers, Jignesh, Sanjana, Rebecca. Great answer. These are my restriction enzymes. RE, commonly we call them RE. RE behave as molecular scissor. They are basically used to cut my DNA molecule at a specific position. Okay, they are very specific in their action. They will not cut anywhere. Okay, there is defined sequence at which they are able to cut. All right, for that purpose we have different type of restriction enzymes. Great, answer is option number 1. +4 everybody. +4 everybody in the last question. Can I see lot many +4s? That was an easy question. Yes, Madhu Kiran? It was an easy question. Can I see lot many +4s in the last question? Yes. All right, great. Next question, kids. Prothallus of a pteridophyte is, okay, we often get confused between Protonema and Prothallus. So Protonema belongs to your bryophytes and Prothallus belongs to your pteridophyte. Okay. So Prothallus of a pteridophyte is, the features of Prothallus, is it multicellular vascular gametophyte? Is it multicellular non-vascular sporophyte? Is it autotropic non-vascular gametophyte which bears sporangia? Is it multicellular non-vascular gametophyte which bears sex organ? Hi Jaswant, hello, how are you kid? Yes, Avinash says option number D. All right. Rest of the kids, come on. Madhu Kiran says option number D. All right. Rest of the kids, come on, come on. Prothallus of pteridophyte is Jignesh, Option number D. Okay. Come on, come on. Rivaka, C. Vanama, Vijay Shankar, option B. All right. Great, 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 great. Perfect, everybody. Prothallus is basically multicellular, having lot many cells. Okay. Yes, Rajya Lakshmi, I'm on one side. Dr. Muhammad. Multicellular, it is non-vascular gametophyte which bears sex organ. Okay. In case of pteridophytes, my sporophytic stage is dominant over gametophytic. Okay, so at first sporophytic stage will come, after which gametophytic stage will there. Okay, in case of bryophyte and pteridophyte, we have haplodiplontic or diplohaplontic life cycle, according to which in the entire plant life cycle 50% of the time my plant is in gametophytic stage, rest 50% my plant is in its sporophytic stage. So the ratio is almost 50-50. That is why we say it is haplodiplontic or diplohaplontic. More specifically, my bryophytes will go for haplodiplontic, since my haploid stage comes first in the life cycle of the bryophyte, and my pteridophytes are diplohaplontic, since sporophyte are the first ones to come in their life cycle. Okay. So if I speak about pteridophytes, sporophyte are basically the first ones, then gametophyte will form or dwell upon it. Okay, so answer will be option number D. Prothallus is my multicellular non-vascular gametophyte which bears sex organ. Alright everybody, so this is it for today's session. I hope you like the session and you will also hit the like button to this particular session. If you have any queries, any question, then do let me know in the comment box. I'll be personally watching and seeing all the comments which are there just after every session. So if you have any kind of doubt left with you, then don't keep it with you, throw it on me. I will surely clear them. And just after the session, in the comment section, do tell me how many questions are correct. How many questions are correct? Okay. This is one more week. Yes, kids. So we'll see in the next classes. Till then, please do hit the like button, share it with all your friends, and subscribe to our beautiful channel. Ved 2025 batch is already live. That is a 1 year long-term course for all the NEET 2025 aspirants. All right, kids. We'll see you in the next classes tomorrow. Same time, 3 to 4. That is time for botany sessions. All right, kids. Bye, everybody. Till then, be happy, be healthy, take care of yourself. See you soon in the next class. Bye, kids. Keep spreading, keep sharing the love, keep spreading the session. Bye, everybody.